I want to give you some background information first about what I'm trying to do. So here's what I'm trying to do. I'm trying to convert lignocellulose biomass, also known as wood chips, leaves, dried up old straw, grass, even moss, and anything else you can imagine. Anything that's pretty much organic contains lignocellulose. So this tree right here uses lignocellulose. Again, it uses the cellulose fibers and then lignin to pretty much connect them together, kind of like glue, and it forms this very strong, durable tree. So cellulose is pretty much just sugar connected to one another. So it's a very long sugar chain. And the goal of what I'm trying to do, and what a lot of researchers have been doing, is try to find a way to break apart that. Because if you're able to break apart those chains, you pretty much have pure sugar that you're able to feed yeast or feed directly to animals, and is a very nutritious, animal feed. If you think about it, everything around us is carbon, oxygen, and hydrogen, right? That's fundamentally what proteins, what, again, carbohydrates, what all these structures and, animal fats, right, so on and so forth, are fundamentally made of. Yes, they have, of micro, potassium and other, chlorine and so on and so forth, right? That but it's a very small percentage. Almost 9 over, I think it's like 95% something is the carbon, hydrogen, and oxygen. And that's fundamentally, if you understand fundamentally, that's all that organic compounds are made from. You can understand that if you have pretty much a supply of organic compounds, you can convert that into anything else you want, right? That's what chemistry is about, is converting one thing to another. And in my case, what I'm trying to do is convert this lignocellulose, which is, regular old wood mass, into something animals can eat. And it's not that too much of a crazy concept. If you think about it, mushrooms and many other organisms already pretty much do this for us. So this is a wild reishi mushroom that is, that literally uses wood in the ground to convert into, food, right? So a lot of horses that you can also think of, and many other grazing animals, have special enzymes in their stomach that convert straw or grass into, food, nutrition, and meat, right? Cows, so on and so forth. But many of these animals, it takes a very long process. And in cows and horses, for example, they have multiple stomachs to allow them to redigest the same mass and eat it to properly break down, because it is a bit of a long process. And many animals like chickens and pigs and so forth don't have that capability or those enzymes to be able to do that. So that's where we need science and processing of the food to be able to make it edible for them. And if you're thinking what I'm thinking, yes, you can use mushrooms to break down the lignocellulose. So pretty much you can get wood chips, grow mushrooms on it, and specifically what you want to do is grow king oysters on them. There's a scientific study that actually shows that you can just grow mushrooms on these wood chip biomass, and feed it directly to the animal, and it's very nutritious. It has proteins, it has, a lot of things that the animals needs. Very, very interesting study. But I would like to do it on a slightly more mass scale, and again, it is something doable, it is something possible, and I do want to go down that route also, but I also want to see how difficult it is to go through the chemical and steam explosion route. So again, remember, what we're trying to do is break apart that lignin and cellulose chain to break it directly into sugars. And one way we do that is through steam. So it's called steam explosions. Pretty much what you do is you introduce a rapid steam into it, and it'll literally explode and break apart those lignin cellulose structures. So I'll show you how I built that machine in a second. And then the second way we could do it is through acid hydrolysis. So this pretty much uses acid to break apart those chains. Pretty simple, but it is a bit of a more technical challenge when you actually have to try to do it. I'll vacuum first. I'll open this up, pull a slight vacuum, try to vacuum this as much as I can, shut this off so this will hold the vacuum, then heat it up and then release the steam, and they'll shoot into the because right here is going to be like 1,000 psi pressure, right here is going to be like minus whatever psi pressure, so going to be pretty interesting to see how this works. You can think of that machine as a very high pressure and high temperature pressure cooker, right? So a typical pressure cooker, and it gets to like 15 psi, but that goes to like 600 psi, so a little higher. Got the vacuum pump hooked up to this. Gonna vacuum, make sure that the air or the wood dust doesn't go into the vacuum, so put extra hose. But yeah, I'm gonna vacuum this chamber out and then heat it up. Alrighty, so here's the contraption. So here's what's going on. Right here is the pipe filled with water. Right here is the chamber filled with wood dust and vacuumed out already. I'm gonna heat it up, give it about like 5 minutes. Just try to heat it up until the pressure gauge goes to around like 600 psi. And yeah, once I heat it up, keep it for that temperature range for about like I said, 10 minutes. Then I'll release the valve. The steam pressure will shoot into here and hopefully crack that. I need to keep it in that temperature again with the steam for another like 10 minutes. So yeah, I'm gonna use this propane tank with a weed burner. Got this free water tank. I'm gonna take it apart, see what's inside, see if there's anything I could use this for a pyrolysis chamber of some sort. Venting it off so I could degas, and it smells like burnt coffee. It's so weird. That is such a weird smell. Oops, that's a little too hot. Yeah, I closed off this valve, so I only have to depressurize half of the system. But yeah. That is a very weird smell. Huh. Here it is. It smells burnt. Probably not a good thing, but it's not burnt. I don't know what the hell that is. Probably like the outside is burnt. Something is burnt. But definitely something did happen. That's interesting. You can like I said, you can smell like the coffee, like burnt coffee. I think what happened was I didn't vacuum it well enough, and there was still some air in there, and it caused it, to burn. Also heated it up a little too high with the blowtorch, not a controlled experiment. But I mean, I think it worked. I mean, again, I have no clue. I have to put it in the yeast reactor chamber and see it out. But so far, yay! It's hard to see, but like this isn't like when I break it apart, it's brown. It's just the edges that got burnt. And as you see, it's completely malleable, right? This is supposed to be a piece of stick, and it's completely flexible. So looks like it did something. I'll put this in the reactor chamber with yeast and grow it out and do a controlled test with just regular wood. And then the third one that I want to do is it's a catalyzed hydrolysis reaction. So pretty much you use like an acid or a base. It doesn't have to be strong. It's very dilute. I think the study I was reading, it was like 0.05% or something, like sulfuric acid or hydrochloric acid. So you pretty much put that in a pressure cooker, and it does way less temperature, way less energy, but you obviously need that catalyst, that acid. So, and then after that you simply neutralize it, obviously with hydrochloric acid. You just mix it with sodium hydroxide, and that converts to salt. So super simple. I will do that next, and we'll start the reaction. Alrighty, here they are. So these are the three things I'm gonna test. This one is the acid hydrolysis. So pretty much what I did with this one was I put in some dilute. Oh, I don't know. I put a splash of hydrochloric acid, muriatic acid in this. Filled up the rest with water. This one was the steam hydrolysis with this thing that I explained earlier. This is just the raw wood chips. And I'm gonna try, growing yeast on these. I'm gonna just use regular old baking yeast. I have some tortilla yeast I want to try, but first I want to just use figure out which one is the best and which process I'm gonna scale up more. But so far, I'm gonna obviously label them. So I'm gonna fill these two up with. This one's pretty hot, or already was hot. It's cooling down now, because I had to after. So what I did was I put, again, I splashed some hydrochloric acid, filled up with water, and pretty much boiled this for like half an hour. and I let it cool down, and I had to neutralize it with some sodium hydroxide, and it started boiling again. So because the reaction it released a lot of heat. So yeah, I'm gonna put in boiling water in these two to kind of sterilize it. Make sure that there's nothing, nothing to cause a spoilage. I'm gonna put a vinyl glove on top just to prevent air from going in and out, and I'll cut a little hole, or poke a little hole in the glove so that the CO2 gases that the yeast ferments can come out. But yeah, so I'll do that process and show you what's next. So just as a taste test, also this is from the this one, the acid hydrolysis. I'm gonna see what it tastes like. It is very, very salty. The reason for the salt is pretty much sodium hydroxide mixed with hydrochloric acid makes salt, right? Sodium, chlorine, sodium chloride, salt. So that's why it's very salty. Before I do ferment this, I'm gonna look up the maximum salinity that yeast can handle. I'm worried this is actually a little too salty. I might have added a little too much hydrochloric acid. If it is, then I'm gonna wash it out or drain it out and just fill it up with regular old water. But yeah, that's it. And as for the taste, tastes like salty wood. I don't know. This doesn't even have it much of a flavor. This one had a pretty distinct coffee flavor. So yeah, let me get to filling these two up and seeing if yeast will survive in high salt environment. So question, if yeast can grow in salt water, Google said no. So had to drain it out twice. So I drained it out, filled it up, tasted it, still super salty. Drained it again, filled it up. This time it's fine. So yeah, all that's left is to put on this and put in the yeast. So that's it for today. This is a pretty simple project, but it's gonna be pretty revolutionary on the farm. Obviously, if you can, turn any sort of bio wood mass, like what's behind me, right? Just regular wood chips, leaves, grass, anything that you can imagine that's organic, and you can convert that into a highly nutritious animal feed. That's kind of revolutionary. Again, there's like the patent you saw, there's like, this is a very old and known technology. I don't even know why they applied for a patent. If you actually check for the patent, they abandoned it because it's a joke to try to get a patent on something that has been known and in existence for decades. But whatever. Luckily for me, they outlined a very detailed process on how to do it. And there's many other research, scientific papers about this. And this isn't going to be something that happens overnight. It's going to take a long process. It's going to have a lot of experimentation that I'm required, but little by little. So I have that. I'm right now waiting on the yeast to finish fermenting. It's probably going to take probably a week or two. I don't know. We'll see whenever it stops the CO2 bubbles. And also, I'm working on a redox flow battery, so I'm gonna have a video on that pretty soon. Waiting on the PVC caps for it, but pretty much everything else is done on that. So stay tuned, and there's gonna be some pretty big stuff on my farm right now. And also the skid steer. Still waiting on the skid steer. Point being, pretty good, pretty fun stuff gonna happen pretty soon on this channel. So stay tuned, subscribe.